Assalamu alaikum. Once again, welcome to my YouTube channel, and today I will be discussing with you the very important topic known as the alpha thalassemia, which is a type of the thalassemia disease. Now, what is alpha thalassemia? Alpha thalassemia is caused by the decreased or absence of the alpha globin genes. This decreased or absence of alpha globin genes leads to the development of the alpha thalassemia disease. Now, talking about the cause, that what leads to the decreased or absence of the alpha globin genes. Actually, what happens that gene deletions occur which causes the alpha thalassemia. So, in the alpha thalassemia disease, decreased or absence of alpha globin genes are observed. What is the cause which leads to the decreased or absence of the alpha globin genes? The cause is the gene deletions. What happens that the healthy genes are completely deleted or removed, which leads to the alpha thalassemia. Furthermore, talking about we know that there are 4 gene copies that are present on the chromosome 16 in the case of the alpha globin genes. 4 alpha globin genes are present on the chromosome 16. We know that 4 copies of alpha globin genes are present on chromosome 16. Now, on the basis of this information, we know that 4 copies of alpha globin genes are present on the chromosome 16s. There are the 4 levels of the alpha thalassemia disorders. Four levels of alpha thalassemia on the basis of the absence of alpha globin gene. Talking about the first level, in this case, if one alpha globin gene is absent, what would happen? One alpha globin gene absent. So the fetus which has the one alpha globin gene absent would be set as the silent carrier of the alpha thalassemia disease. So the baby which has the one alpha globin gene absent would be characterized as the silent carrier of alpha thalassemia disease. What does this mean by the term silent carrier? The term silent carrier means that this particular baby would not express the symptoms of the alpha thalassemia, but it would carry this alpha thalassemia disease to its offsprings. Second level, if 2 alpha globin genes are absent. So if the 2 alpha globin genes are present in the fetus, that fetus or baby would be said to be as the carrying the trait of the alpha thalassemia disease. So that particular baby, if having the 2 alpha globin genes absent, would said to be carrying the trait of alpha thalassemia disease. Carrying trait of alpha thalassemia. Now talking about the third level, that if if three alpha globin genes are absent, if three alpha globin genes are absent, so if three alpha globin genes are absent, it would mean that the baby would be carrying the disease known as the hemoglobin H beta 4 disease. So that baby would be carrying the disease known as the hemoglobin H beta 4 disease. Now discussing about the fourth level of alpha thalassemia. If 4 alpha globin genes are absent, if 4 alpha globin genes are absent, so if 4 alpha globin genes are absent, that baby would be said to be carrying the disease known as the hemoglobin Bart gamma 4 disease. The person would be carrying the disease known as the hemoglobin Bart gamma 4 disease. Now talking about in this alpha thalassemia disease, what substances gets accumulated? So accumulation of substances during the disease known as the alpha thalassemia accumulation of substances HbA number 2 beta aggregates means Hb hemoglobin H beta 4 number 3 hemoglobin Bart gamma 4. So these three substances gets accumulated during the alpha thalassemia. HbA beta aggregates of the hemoglobin H beta 4 and hemoglobin Bart gamma 4, these three substances get accumulated during the alpha thalassemia. So I have given you the brief description on the disease known as the alpha thalassemia and hope you have understood it well. Last but not the least, don't forget subscribing MS Lectures. Thank you.